Mitosis versus Meiosis by Grant Jackson, Emma Perkins Johnson, Vanessa Van Pelt, and Tom Higgins.
Interphase consists of G1, where the cell grows, the S phase, where genetic material duplicates, and G2, where the cell prepares to divide. This phase takes as long as 90% of the cell cycle.
In prophase, chromatin starts to wind together. Sister chromatids are created, and centromeres start to form between them to create chromosomes. As prophase progresses, the nuclear envelope will start to dissolve. Also, centrioles and spindle fibers will start to attach to the centromeres. Metaphase is the aligning of the chromosomes. The condensed chromosomes, DNA wrapped around the proteins, align equidistant from each centriole. The spindle fibers will help to get the chromosomes all in one straight line before anaphase begins.
In anaphase, the sister chromatins are pulled apart. The spindle fibers help to push away the chromatids to opposite ends of the cell. This picture shows two chromatins being pulled apart. Telophase is the final phase in mitosis. Each opposite end of the cell has a set of 23 homologous chromosomes, each side containing one set of the original pairs. The nuclear membrane will then start to reform. Kinesis finishes off the end phase, also known as the mitotic phase. During this process, the cytoplasm will be split in half, and this will conclude the process of cell division, ending with two diploid cells.
The paired chromosomes, now consisting of 4 chromatids, are referred to as tetrads. Next, the tetrads exchange some genetic material in the process known as crossing over. The tetrads move to the middle of the cell and line up across the spindle. Then, homologous chromosomes will start to separate. Finally, in telophase 1, the chromosomes arrive at opposite poles. Each pole now has a haploid daughter nucleus because it has only one set of chromosomes, even though each chromosome consists of 2 sister chromatins.
The steps of meiosis 2 are very similar to the steps of mitosis. The difference is that instead of starting with a diploid cell, meiosis 2 starts with a haploid cell. A haploid cell is a gamete which has a single set of chromosomes. This can also be shown by N, which also means 23 chromosomes. In prophase 2, spindles will start to form and attach to centromeres. Then it will move to metaphase 2, and this is where the chromosomes will then start to line up in the middle of the cell. And moving into anaphase, this is when the sister chromatids will start to go to opposite poles of each of the cells. The end result of meiosis 2 is 4 haploid gamete cells.
In mitosis, there is 1 division, increases in genetic variation, somatic cells, 2 cells produced, genetically identical cells, healing damaged tissue, and tissue growth.
In meiosis, there are 2 divisions. It happens in gamete cells. 4 cells are produced, and it has metaphase 1 and metaphase 2. Both mitosis and meiosis include interphase and DNA replication.